the active value has to be even integer. Okay, so essentially we have a 0, 0, 0. This is not. Yes. Yeah. Oh, sorry.
I think we can start. So, good morning, everybody. It's a pleasure to host in this virtual meeting Gerhard Hummer from the Max Planck Institute of Biophysics. His talk will be about molecular simulation in the times of COVID, and we look forward to hear your presentation, Gerhard.
Well, thank you. Can you hear me?
Yes.
Yeah, good. So thank you. First of all, a huge thanks to the organizers up front for holding this meeting. I think this is very important for all of us, for the community, and I'm really excited to be part of this. So as you can see from the title, I very much acknowledge the fact that all our lives have changed, and as I will show you also, our research has changed, and we have in my group and my collaborators groups, my friends groups, many of us have been trying to contribute in a meaningful way to the fight against the COVID-19 epidemic. So just here on my first slide, you can see an outline of the virus life cycle from a recent review by Brett Clownsinger. And as you can see, this is a pretty complicated life cycle that can be, however, divided into a few key steps. The first one is the key part of the infection, the entry of the virus into the cell. And when it establishes itself, it leads to the translation of its genome and the viral reproduction. A key step then is to assemble new virions, and in the last step, to release them to the outside. And so in my talk, I will show results from our work involving molecular dynamics simulation, where we looked at two key steps in this viral life cycle. The first one in presentation is an element that is very common to viral infections, and that is immunosuppression. And as it turns out, that is associated with a protein, a protease specifically, that is part of the translation and polypeptide processing machinery of the virus. And then in the second part of my presentation, I will show you also results associated with the entry of the virus. And specifically, we will be looking at the so-called spike protein or S protein on the viral surface, with which SARS-CoV-2 attaches itself to the host cell receptors. So first part, I will concentrate on the so-called Papain-like protease. This is a protease that has multiple functions, and this, of course, is common in this very dense viral genome that proteins do a bit of moonlighting. So the papain-like protease, it as its name implies, it cleaves protein chains, and it does that in part to support the processing of the multi-protein chains produced from the viral genome and cutting them into functional protein units. But in addition, the papain-like protease also is very actively involved in the suppression of the innate immune response of the host cell, which of course tries to inform the outside that it has been infected and activate antiviral responses that, for instance, can kill an infected cell. And this would be, for instance, in this case, the type 1 interferon innate immune response. So this is work that has been done in collaboration with the group of Ivan Dikic, who is at the University Hospital in Frankfurt and also a fellow at our institute, and with his then postdoc Dong-Hyuk Shin, just took up a faculty position in Korea. And what they found was extremely interesting. They found that this papain-like protease, which was actually known from earlier work, was cleaving polyubiquitin chains, and it was, but also it was cleaving another ubiquitin-like protein, ISG15. But what they found was that the new SARS-CoV-2, had changed its catalytic activity from favoring the cleavage of diubiquitin or ubiquitin chains to cleaving this ISG15 ubiquitin-like protein. And that was very interesting because cleaving of ubiquitin and ISG15 are both associated with modulating immune responses. So what our collaborators then could show is that the driver for this transition from cleaving ubiquitin to cleaving ISG15 was a change in the affinity, the binding affinity. So in a equilibrium binding experiment measuring the KD, the dissociation constant, they found that the KD had inverted. And this is shown here in this plot on the right, where you have now much stronger binding to protein ISG15 and weaker binding to K48 linked diubiquitin. That is the driver for this change in the kinetics. So then you can of course ask, what is this good for, and why does it change? So we know from many years of viral studies that infected cells often activate this ISG15 production, and they attach ISG15, a diubiquitin-like molecule, covalently to many proteins of the cell, as well as to the proteins produced from the viral genome directly. But it's actually not very well understood what exactly this does. And there's a number of theories out there for what ISG15 modifications do. They could alter the oligomerization states of proteins, or also modify cellular localization of these proteins. And what we now found, or our collaborators found, is that SARS-CoV-2 activity decreases the isolation of cellular proteins. They could show that with proteomic studies. And one of the key proteins involved in this innate immune response, but we have a better understanding of what isolation does, is this interferon regulatory factor 3. And so just very briefly, in a nutshell, if it's modified by ISG15, covalent attachment, then gets phosphorylated, and that leads to uptake of this IRF3 factor into the nucleus, where it activates the antiviral response by acting as a transcription factor, like protein. And so this, however, is altered if ISG15 is removed from IRF3. Then IRF3 is degraded, not taken up in the nucleus, and that leads to a suppression of the interferon response. And so that's, in a nutshell, what we think happens in this particular case. But I have to emphasize, this is a much broader response that is happening here. So what is going on then at a more mechanistic molecular level? For that, Dong-Yuk Shin, a postdoc in the department of Ivan Dikic, solved the structure of the complex between PL-Pro, the protease, here shown in pink, and the diubiquitin. And what we could see in the structure was that there are two changes between SARS-CoV-2 and SARS-CoV that are interesting and could contribute to this shift to a immunomodulation activity rather than ubiquitin processing activity. And that one is a change from a leucine, position 76, in old SARS to a threonine, position 75. And then there is another shift with the opposite change in polarity from a serine to a valine. So this, of course, got us very interested. And Laura Schultz in my group, and together with Reza Medipur, they set up MD simulations of various constructs. And what they found consistently was that the ubiquitin actually bound to the SARS-CoV-2 PL-Pro is unstable. So on an MD timescale, which is quite unusual because protein-protein complexes, they're rather difficult to simulate, but consistently what we found was that the ubiquitin started to dissociate. So this is illustrated here in this RMSD versus time plot, where we can see that in 4 out of 6 simulations on a microsecond time scale, the ubiquitin comes off. This is indicated by the orange and yellow hues. By contrast, ISG15 remains stably bound to the protease, so indicating, yes, indeed, it is a good substrate of this protein, whereas diubiquitin is a weak substrate and has a much reduced binding affinity, consistent, at least this level, with the experimental observation of a shift in KD and a reduced catalytic activity for ubiquitin. So, but we had to do some controls, and this is one important control is to introduce the mutations or these changed amino acids between SARS-CoV and SARS-CoV-2 back into the old SARS-CoV background. So we take the new valine and then use the threonine residues, and we make mutations in the old SARS structure. And what we found was we didn't, in this one simulation, see full dissociation, but the interface clearly opened up, and then in one of the intermediate forms that we had seen before was an important step towards protein-protein dissociation. So one of the wonderful things with MD simulations is that then you can start to zoom in and you can see what is happening at the very detailed, at the near atomic level of these processes. And what we found was actually quite interesting, that ultimately the main driver that we could see in the MD simulations that explains the difference between this substrate preference of the protein-like papain-like proteases is a small change in the hydrophobicity. It is the difference between the threonine here and its interaction with the isoleucine 44. What happens is that increase in the hydrophobicity of the interface between threonine on the protease and the isoleucine on the ubiquitin can transiently lead to an accumulation of water. The more hydrophilic character lets water enter this two amino acid contact, and it is this consistently water entry is what leads to dissociation. Just as an important point to show that mechanism is not all we can do with MD simulations, I want to emphasize that this MD is also a good tool leading us forward in the development of new therapeutics. So what our collaborators started out with is an inhibitor that had been developed for the papain-like protease in the Mesecar lab almost now 12, 13 years ago. And this is for the old SARS. And this inhibitor, as it turns out, also works very well for the papain-like protease of the new SARS. And we could show why this is the case by docking it into the new SARS protease structure, and could find that indeed the binding pocket is very well conserved, and the inhibitor binds very stably in a mode that is essentially consistent with the old SARS. And that, of course, opens up now a window of opportunity to target this binding site as a possible way to suppress the viral proliferation. And indeed, that's what our collaborators are doing. And at a functional site, what they could show is that that this inhibitor reduces the protease activity, and they could, the lab of Sandra Cizek at clinic in Frankfurt could also show that with this inhibitor, you could actually suppress viral replication and viral proliferation in live cell assays. And that should show one result, looking on the right here, looking at the mRNA level in the indicating the level of infection. So we have both mechanistic understanding and with at least a first inhibitor, a route forward to targeting this process and hopefully deal with replication rates. But maybe even more interesting from a intervention perspective, of course, is the development of vaccines. And most vaccines look at proteins at the viral surface, and in particular at the so-called spike protein. This is shown here in a cartoon of a viral entry. So here, this circular particle indicates the SARS-CoV-2 virus decorated with these S proteins, these flower-shaped structures that bind to the ACE2 receptor on the viral on the host cell surface, and that leads to either direct entry through the plasma membrane or, and again, the process that is not so well understood, entry through the endocytic pathway and then endocytic escape. So we started modeling this SARS-CoV-2 spike protein or S protein already in the first weeks of March. This was work pushed forward by Matt Sikora, but then really involving a very big part of my group and my department. And you will see the names listed later. So what we knew at the time was that we had a pretty decent electron microscopy structure of the head of the spike, but the rest was essentially not known. There were no structures. So we pieced them together using various modeling techniques and using information also from the old SARS. So we put together a stalk model as well as a model of the transmembrane domain and the membrane anchors. And importantly, we included glycans to decorate the surface of the spike protein based on extensive mass spectrometry studies from several groups, particularly the group of CRISPR. And so built a full-length model and inserted it into the membrane and then set up an MD system. What is maybe interesting from a practitioner's point of view is we decided to put 4 spike proteins into the membrane. Now, you might think this is a bit crazy because you already have an enormous size system. So now we have it almost 4 times as big. However, there are certain advantages of doing this. One is that with only 1 spike, now we have big problems with self-interaction across the periodic boundary. With 4 spikes, we have, of course, 4 times the cost, but we also have 4 times the sampling. We ended up with a system with 4 million atoms. To make it run a bit faster, we used heavy water that allowed us to use a 4 femtosecond time slip. And in this way, we could collect about 2.5 microseconds of ND, which by having 4 proteins amounts to about 10 microseconds of aggregate sampling. And here you can see a movie. I don't know if it moves on your screen. It indicates spike is actually sitting quite flexibly on the viral surface. And that is illustrated here in more detail, where we emphasize that the joints in the viral stalk that we termed hip, knee, and ankle with head or body, upper leg and lower leg forming the more rigid parts of the protein. When we zoom in on and do an RMSD alignment on these more rigid parts, you can see that the rest of the protein really frays out. These are superpositions of structures along the MD trajectory. And this indicates these are very flexible hinges, and that's also emphasized by this distribution of the hinge bending angle. That was very interesting. However, as we were doing these ND simulations, we learned that in our institute, a data set was collected by the group of Martin Beck, who's at the Institute and at the EMBL, in collaboration with the lab of Giacomine Locker at the Paul-Ehrlich Institute, whose group has been doing the virology. And what the data set that was collected is a very high quality cryo-electron tomography. And you can see here a cut through the 3D volume of this EM tomography data set, where you could maybe see the viral particles as they appear. And here you see a cut illustrating them. And on the black dots on the surface are these spike proteins, and they were beautifully resolved in this data set. And remarkably, already in the raw tomogram, they could see the hinges, and not just the spike body, but you can see the hinges, and you can see that the stalk actually is flexible and that it bends. And that would also explain the irregular structure of the protein on the viral surface. What we could then show was, by comparing notes, is that our MD structure, molecular dynamic simulation structure, could be just placed directly into the raw tomograms. These are not flexible fits. You may have seen flexible fitting of MD structures into electron density maps. Here, we just took snapshots from the MD simulation. We took these extremely high resolution tomograms without any further processing, and you can see the MD structures fit very nicely into the structure. And I'm not going to discuss that we also have very good agreement with the subtomogram averages at a very high resolution. I just focus on this aspect and emphasize now this beautiful consistency here also in this central view graph, where you can see a bigger part of the viral surface taken from a raw tomogram, now decorated with MD snapshots. Now, why do we think that the stalk is so flexible? Well, we think that a very possible explanation is that this increases the avidity, and the avidity is a measure of avidity refers to having multiple interactions in parallel, to each one of them relatively weak, but jointly making the interaction very strong. And I think that by having flexible attachment points, it's easier for the virus to have multiple interactions with spatially spread out ACE2 receptors on the human cells, tightening the interaction and thereby leading to a more effective infection process. So one final point I would like to emphasize, where we saw not only nice correspondence, but between experiment and the simulations, but gained additional insight, concerns the glycosylation of the spike protein. So these viral proteins are covered by an extensive protective shield of sugar-like molecules, and here on the far left you can see these are large glycans on the surface, and they fit very nicely into the electron density, both the single particle EM structures are shown on the left and on the tomogram shown here in the middle. And even in the raw tomograms, again, you can actually see very nicely that you have a strong additional density decorating the surface of the protein. And so here you see now what this does on the basis of the MD simulations. When we average the density of the glycans on the viral on the spike surface, plotted here in green, you can see that these large and very dynamic, very floppy sugar chains effectively mask almost the entire surface of the protein. They expose very, very little, just primarily the receptor binding domain with which it interacts with the ACE2 receptor. And what that means is this is a very effective shield against the binding of antibodies. So it's effectively the virus uses this coating by glycans as a means of giving antibodies very little distinct epitope surface to bind to. So this brings me to my summary slide, and I hope I could give you at least an idea of how molecular dynamics can be useful, can make meaningful contributions in rather difficult times for all of us. The first part of my presentation was focusing on an interesting and I think a very important aspect of what viruses make such highly effective machines and so successful in combating our immune response. I showed you that in the case of SARS-CoV-2, as compared to the original SARS or old SARS, we had this shift from a deubiquitination to a deisolation activity. And the interesting point, maybe from an MD and biophysics perspective, is that ultimately this shift and the greatly enhanced ability to mess up our immune response is that 1, 2, maybe 3 water molecules can more easily penetrate into the otherwise hydrophobic interface of the protease with ubiquitin, and which does not affect the ISG15 interface. Conversely, an increased hydrophobicity, by adding this valine, strengthened the interface with ISG15. I also showed you that MD simulations can help us when assessing and hopefully improving inhibitors that target this process of the protease. I showed the results for the GRL0617 inhibitor developed by the Mezicar lab originally for old SARS, which also stalls viral reproduction for new SARS. Then I showed you results for spike, the protein at the viral surface, and these simulations revealed hinges in a spike stalk, and then in completely independent electron tomography experiments, these were recapitulated. And I concluded by showing the dynamic glycan code, and which is consistent with what we see in the in situ tomography, and I think which is very important. That's something we're still actively working on. There's a first paper submitted now on looking at our ability to still find good epitopes for antibodies, how that can lead to a strong and robust immune response. For that, I would like to thank my people in my group. So the PL-Pro protease simulations were carried out by Laura Schultz, a PhD student in my department, and with a lot of help and support and insight from postdoc Reza Medipur. And I really cannot thank enough our experimental collaborators, Dong-Yuk Shin, who just left Frankfurt, is now starting his faculty position in Korea. This has been a fantastic collaboration with the lab of Ivan Dikic, and I really look forward for more of this work. And also we got from the virology help, from a lot of help and support from Sandra Chisek. The tomography experiments that I showed were carried out primarily by Beata Toronova with help of the people in the EM facilities in Heidelberg at the EMBL and in Frankfurt. And it's been a fantastic collaboration with the Martin Beck group at our institute and still at the EMBL. The virology was done by Christoph Schürmann in Giacomini Kreinzer Locker groups, again with lots of help from other people at the Paul-Ehrlich Institute. And finally, the ND of the spike proteins was spearheaded by Matt Sikora, and the model building and ND simulations got a lot of support. It was a huge team effort by Søren von Bülow, Florian Blanc, Michael Guest, and Roberto Covino. We just started his independent group at the Frankfurt Institute for Advanced Studies. And with that, I would like to stop and take your questions.
Thank you very much, Gerhard, for this very, very interesting presentation. I'm sure there will be lots of questions. So there is one in the chat. Please comment on how you would mimic the effect of different environments like extracellular space, cytoplasm, and inside nucleus.
Yeah, this is, I think, a very interesting and important question that, in particular, when I think about the spike protein, where I just briefly mentioned that it's not so well understood how, at the moment, how the entry of the virus really happens, whether it is directly to the plasma membrane, and there's papers describing that, or through endocytic pathways. And these environments are very different in their redox levels, but in particular also in terms of pH and salt conditions. And so it would, at the moment, we took, for the environmental conditions, we took a neutral pH type of setup, but clearly for fusion with the in part of endocytic escape, we would probably have to revisit some of these initial protonation analyses and maybe even change them. In addition, there is, of course, also the question, not just the environment, but of the protein itself, where also many things are not so well known, and that concerns in particular cleavage state. So when the proteins actually get actively remodeled by host cell proteases. And when exactly these transitions happen is again, so cleavage, for instance, of spike head is not so well understood. So interesting questions and lots to do. We took a first stab at this, but I think we will be busy with this for times to come.
Thank you, Gerhard. This was a question from Strabonit out of India.
Okay. Now we have a question from Stefan Wolf.
Yeah, hi, Gerhard. Thank you. It's a very beautiful talk and great work that you did there. Concerning this breakup of the interface with the ubiquitin by a single amino acid, that's quite interesting, really. Did you have a closer look at what is actually modulating? So what this one single mutation is doing to break up a complete protein interface? Because that seems like a huge change in free energy between the for binding, just but based on such a small perturbation, it's quite surprising.
Yes. So I don't, at the moment, we don't have any free energy results. So obviously this would be a huge effort. And so the only thing that we see is kinetically, the main driver is, I mean, it's a very small interface to start with. The affinity is weak. It's in the 10 micromolar KD to start with. So it's not a strong binder. And I think that's what allows this transition in bound and unbound state to happen at all, right? Because for a strong binder, it would not happen. And in all we have 4 dissociation events out of 6 runs of 3 microseconds, or 30.2, I think. And in all 4, what we see consistently is that water comes in, this threonine isoleucine interaction, and so you lose, effectively you weaken 1 out of 3 hydrophobic contacts, and I think that is enough to make this a weak thing. Now, I should point out that this is a diubiquitin, so the other ubiquitin sits in the active site. That is still tightly bound, and I think so that's why we don't see a complete dissociation. We just see one ubiquitin come off or come partially off. I think maybe as a bit of explanation. That's why the overall binding is still, I think, even in the experiment, it's now reduced by like 30-fold or so, but not 1,000-fold.
Okay, thank you.
Mm-hmm. So I think, I think that maybe 30-fold decrease would be quite consistent energetically with effectively losing one hydrophobic contact.
Now we have a question from Daniel Madulu.
Thank you for the nice talk. I have a question on the role of water. In one of the slides, you showed that water was able to penetrate at the hydrophobic interfaces. So how did it also affect the binding of the small molecule? So I need more, maybe explanation, maybe I missed something. Thank you.
Yeah, so at this interface, right, the water, the role of water, it forms additional hydrogen bonds with the threonine. For the small molecule inhibitor, there we did not specifically look at water there because it was very stably bound and really quite consistent with the experimental structure. And just to maybe add to that, the inhibitor is interacting both through hydrophobic interactions and hydrogen bonds, as well as charge interactions with the protein. So their water did not, they haven't examined it in greater detail, but water did not play a particularly important direct role.
Thank you.
Mm-hmm.
Now there is a question from the audience. In case of sensing devices for tracking infectious person, are there any glycans or sugar that would be important to be detected?
So I don't think I would be the right person to answer this. I can just maybe give it a try. The important thing, of course, is all the glycans are our glycans, and that's one of the reasons that the virus uses this. So it uses the same sugars that we add onto our cell surface receptors and other proteins that are on the outside of cells, and it hijacks the glycosylation machinery as well as the sugars from the infected cell, and uses that. Of course, makes it so difficult for the immune system to deal with these enveloped viruses, because much of their surface is covered by the same chemistry as our own cells. And so antibodies actually have little chance. And I think now, with respect to the question, that also means the sugars per se would probably not be particularly useful. However, if you look at maybe protein fragments with sugars, that might very well work. But again, maybe not the right person to address that. But still an interesting question.
And then another question from the chat. Can you comment about the differences between locked and open structure of spike protein regarding its entry process into the host cell?
Yes. Yeah. So this is a very important question where we had lots of discussions about. There are several structures of the spike head out that are either completely closed or they have parts opened up, and it is thought that interactions with the ACE2 receptor in particular require quite a bit of opening, and also some antibodies bind only to open or only to the closed state. In our simulations, we therefore used a mixed structure with one subunit open. In the experiments, what we found was, which was surprising, that in our colleagues' viral preparation, the majority of spike subunits was in the closed state. Only upon closer inspection, we could detect some open state structures. At the moment, we don't have a good way of looking at this close to open transition, but that again would be, I think, a very important and very exciting project. And I also hope that from the tomography as well as from single particle EM studies by other groups, we will get more guidance on how this transition happens, what induces it, and also what are the factors setting the equilibrium. Again, could also be something that came up in the first question, environmental factors such as pH and salt conditions.
There is another question from the audience regarding the force field that you use for the simulation.
Yes, so these simulations were done with the charm force field.
Okay. So I have
the spike simulations. Yeah. What?
One last curiosity myself. If this flexibility of the spike is so important for the avidity and probably also for the infectivity of the virus, is there any data that you compare maybe with the previous SARS? Is it known how is the flexibility in that case?
Yeah, so we think that old SARS also was flexible because the stalk is essentially 100% conserved. It's among the most conserved parts, in part because in the post-fusion structure, which is the stable structure of so we can think, or at least I tend to think of the pre-fusion structure that I simulate as a metastable structure, like spring-loaded for a transition into a more stable structure, post-fusion. But other envelope viruses seem to have much shorter stalks and more rigid structure, but we do not interestingly, also for other viruses, we do not know very much about these elements because yeah, you do need very high resolution structural studies like EM tomography to be even able to see these things to the degree of flexibility. So we are thinking now, of course, of ways of looking into this in more detail. And one possible way would, of course, be to capture infection events tomographically, or maybe also with high resolution, super resolution light microscopy to see if indeed this is a single receptor binding event or a multi-receptor binding event where you have recruitment. So it'll be interesting to see that. But this is, at the moment, I can only emphasize this once more. This is speculation. We just do not know. We just find it interesting as a possible explanation for why this is there.
One last question. I would like to know if there is a specific reason to put heavy water in the simulation.
So this is, of course, something that concerns all ND. I mean, the reason that we include the water is that first of all, it's mechanistically may be interesting, but the force field descriptions require that so-called implicit solvent models that do not include the water are just not as well calibrated at the moment. So we probably could not have the same level of accuracy and detail in those simulations, because these interactions between the proteins, with the membrane, and within the protein and so on have to be very well balanced, and with water, they're there. By using 4 spike proteins relatively densely packed on the membrane, we in effect try to minimize the amount of computation time we spend on water. But if, as the person asking the question clearly realizes, this is still in many ways looks like a waste, but I think at the moment it is largely unavoidable. And as I also showed with the protease, functionally sometimes very important.
Okay. Thank you very much again for this nice presentation. And I think we have a coffee break now, and we reconvene at 10:15.
Thank you. Goodbye.
Bye-bye. Hi. We wait a bit more, but I see many participants are not in the chat yet.
That's good.
Okay.
Alessandra, should I put my presentation in share mode already? Or?
Yes, I think you can start. You tried already, so I think it's.
I think it should work, but of course you never know.
You look experienced with this type of talk.
Yeah, but you know, I have this bad karma that I always get a problem with the software updates exactly the same day I'm supposed to give a talk, which is, I mean, it's remarkable.
Me too. The first time I was speaking.
Yeah, yeah. But no, I think that Zoom usually works rather well. I mean, I think in this type of things, yeah. I guess it's better, or should I? No, it's too much light, I think, yeah. I think it's better with those, yeah. Down.
We wait 1 more minute, then we start. Well, it's a pleasure to introduce Professor Ville Kaila from the Department of Biochemistry and Biophysics of Stockholm University. And his talk will be elucidating mechanism of biological energy conversion by simulations across scales. Thank you.
Great. Thank you so much, Alessandra, for the nice introduction. And thanks for the organizers to, first of all, organize this workshop and have it on Zoom despite of these big challenges. So I thought today I would like to present to you a challenge in the simulation world, a system that we have been working on for a very long time. And basically the outline of my talk is the following. So I will start by telling about a structural revolution that has happened, I think, all over in biochemistry. I would then go on to tell a little bit about the multi-scale approaches that we use to study these type of biological questions, and then introduce to you a very interesting molecular machine, the respiratory complex 1 machinery that catalyzes a fascinating 300 angstrom long charge transport process. with that, we could start thinking on the motivation of this work. And at least for me, what I think is truly fascinating, that all organisms, irrespective of their complexity, if they're very simple singular organisms, more complex beings like humans, they of course need an input of energy to grow, to adapt, and possibly move in their environment. And of course, you couldn't do this without some input of energy. And in nature, this can take place in two various forms, either in form of light energy or in form of a chemical energy. And of course, as you know, any process in biology takes place by enzymes. And also enzymes are the key catalytic players in catalyzing capture of either the chemical or light energy. What I think is truly fascinating is that on a chemical level, these processes take place by elementary particle transfer, by mainly coupled motion of protons and electrons. And we have two main such cycles in nature. We have the photosynthetic one, where light energy basically drives oxidation of water molecules to protons and electrons. You take the electrons, combine it with inorganic molecules like CO2 in the carbon fixation process. You build more complex organic molecules like sugars. And then you take basically these high potential energy electrons back and combine it with something that's a good electron sink, for example, dioxygen. And for that, you need some protons as well to restore, basically get water back. So at the heart of energy conversion, you basically have a counterplay between these protons, electrons, and in light activated processes, light energy. So there's been a wonderful structural revolution in biology in recent years, and this has helped us, of course, understand also the molecular machines or provide blueprints for them. And one very important development is the cryo-EM technique, which is today resolving almost standard-wise structures at around 3 angstrom resolution, but even better. And we now have all members of, so to say, the mitochondrial respiratory chain. We have also photosynthetic system, but also many light-driven ion pumps. And this, of course, provides us a starting point to understand their function. But of course, it's not fully trivial to couple together the structure and the biological function. And to this end, of course, we would like to zoom in, and we can do that thanks, I think, to the very nice computational methods that provides a very powerful way to ask mechanistic questions about the system. And this has been introduced many times on this workshop already, but maybe I just highlight a little bit the philosophy that we are using in my lab to study this system. So to explore enzymes, you of course need to treat the chemistry. And for treating the chemistry, of course, then the quantum chemical methods provide a very powerful toolbox. And we use very much the density functional theory methods in my group due to the good scaling and accuracy. But also for some problems, we try to go to more, so to say, better methods, correlated quantum chemical methods that we then couple together via the QMMM framework to methods that are good for exploring the phase space of the system. And here we use widely atomistic molecular dynamics simulations, but also free energy methods of various kinds that were very nicely also introduced during this workshop, and even up to a, so to say, coarse-grained representation of the system to get interesting information on the longer millisecond time scale. So how this could work is that we, let's say, starting from an experimental static structure, run an atomistic MD simulation, we propagate the system on some microsecond timescales in various states. And then we open up parts for quantum mechanical treatments for a structure, an ensemble of structures. For example, here is an ATP molecule being cleaved by a proton transfer reaction. And for this, we can then use some methods for example, to calculate free energy profiles, I don't know, with umbrella sampling or something like that, and then couple various specific conformational changes, let's say, in the protein to differences in activity that we can read out from these free energy profiles. And this, of course, provides a very powerful way to design experiments and validate your mechanistic understanding experimentally. So bioenergetics, which we're interested in my lab, basically ask the question, how do you, from a given, let's say a chemical reaction or light-driven reaction, very often in a membrane protein, how do you use this free energy? How do you transduce it to transport ions, for example, protons or other type of ions across the membrane, such that you prevent possibly kinetically the backflow of the ions in the wrong direction? And of course, you need structures for this, and here we rely very much on cryo-EM structures and crystallography, and of course the methods of computational biochemistry then provides us as the details to ask how this could happen in practice. But what we also try to do very much here is based on the simulation, design, for example, mutagenesis experiments to perturb the system and test them by different biophysical experiments, how this would perturb the activity. And we have also started using a little bit of an protein engineering and artificial protein design approach to ask and, so to say, isolate various catalytic parts of the system into a simpler environment. And of course, the downside of this is that it's not enough that you only have supercomputers and a computer lab, but if you want to run experiments, you also need to build an experimental lab, and this is the picture of the experimental way in our lab that enables us to test some of the mechanistic hypothesis. So let's go to the biological background a little bit and ask, how does this enzyme, this complex 1 enzyme that I would like to introduce to you, how does it power cell respiration? Cell respiration takes place at membranes basically, and what I show here is the inner mitochondrial membrane, and it has these embedded membrane proteins in it. Basically, what they do is that they take electrons from the foodstuffs that we eat and then transfer to these various centers and lower basically release energy stepwise. And this energy release is coupled to motion of protons across the membrane. And of course, this couldn't happen if we wouldn't have an electron sink. And this is why we breathe oxygen. So most of the oxygen that you breathe goes to this process. And that basically then takes these electrons from foodstuff to drive the process. And of course, this proton pumping across the membrane generates a electrical gradient across the membrane. And it's fascinating actually that across this 30 angstrom thick membrane, you have a voltage difference of 0.2 volts. So it's massive, basically the forces talking about on so small scales. And then the protons, what they would like to do is basically go back to the other side of the membrane, and this you can then couple to, for example, synthesis of ATP molecules or active transport. And let's now focus on this first initial member of the complex. It's called complex 1, and it's a very big enzyme, a PCET machinery, coupled proton coupled electron transfer machinery. Is basically up to 1 megadalton in us humans, and we now have structures of this molecule from several groups solved in recent years. And it then functions as an initial electron acceptor in this respiratory chain of both mitochondria and bacteria, and it transfers electrons first from a general cofactor NADH. This happens by a concerted, basically a H- transfer, so coupled proton and electron transfer move through these aromatic ring systems, and then the electrons continue to a wire of around 8 iron sulfur centers to this quinone molecule over here. And maybe you have seen quinone in cosmetic products or something like that. That's basically because it's a good electron scavenger, and it might kind of prevent you from becoming wrinkly with time. Good. And then basically the quinone is reduced to a quinole molecule here in the active site. And this fascinatingly couples the proton transport up to 200 angstroms away from the active site. So this is really a long range coupling process. This is highly efficient, such that you can turn around this machinery and run it in reverse. You can use a pH gradient to extract electrons also from a quinone molecule the other way around. And it has basically everything that there should be for microscopic, like textbook thermodynamics. There is a microscopic reversibility such that if you perturb one end of the molecule, you will also measure an activity change for the quinone oxidative reductase function. This is exactly how it's supposed to be, but of course the question is how this could take place. One could also say that it's of high biomedical relevance because mutations linked in this enzyme are related to half of all human mitochondrial disorders. And here is, you can pick a favorite of the disease. It doesn't necessarily mean that the disease is caused by mutations in this enzyme, but it has a link. And one possible explanation to be related to this is that under some conditions, electrons leak out from this enzyme, and they of course cause then reactive oxygen species and harmful effect in the cell. So what we would like to understand is how can this machinery basically have couplings across such big distances? What is the mechanistic principle of such system? And I would like to divide this talk into key questions. I mean, first to ask, how do the electrons enter this machinery and trigger the proton pumping? Where does the coupling between the electron and proton transfer process happen? How are protons pumped across the membrane? I would like to highlight some conformational changes related to this machinery. And then if there is time, I could also show to you a very interesting modular twist where this machinery is used in photosynthesis to do a very, very important process over there. So since this is a workshop, I thought that it could be interesting for this community to have some thoughts on how to model such a complex system. And basically, I mean, of course, similarly to any other maybe MD study or computational study, you need a structure. And we have worked with various type of complex one structures, from bacteria, eukaryotic, but also photosynthetic systems. Solved in recent years. Then this is a proton pump, so you need to concern yourself about the protons. This protein has more than 1,000 titratable groups, and we usually aim to assign protonation states by a Poisson-Boltzmann electrostatic approach coupled to a Monte Carlo sampling of the different states to get at least an idea of good starting conditions in the simulations. Then basically we need to model the substrate quinone and build a membrane. And for this we use usually a 3-component membrane with PC, PE, and cardiolipin, mimicking the mitochondrial inner membrane. And then basically we need to build water and ions around this. And then unfortunately you end up with a system with around 1 million atoms in this. We're doing the modeling with the CHARM36 force field, and we use regular water instead of D2O. That seemed to be a very nice idea that the previous speaker talked about. So we use H2O. And tip-trippy water molecules, which is not always the best, of course, in systems like this. In addition to not doing simulations only in maybe replicas for one system, we try also to get an understanding of the various kinds of complex ones. And there are the result species from bacteria and mammalian systems and yeast to other variants of the system. And we think that by modeling all of these, you get basically, you can understand better the functionality. In order to model part of this process, which is the motion of protons and electrons, you of course need to be able to parameterize them also to describe them classically. And to this end, we have developed force field parameters for, so to say, the iron sulfur centers, the flavines, the NADH systems, and quinones in various oxidation states. And these are very often based on density functional theory calculations, where you basically then can derive these force field terms to be able to model this process on a classical time scale. But then to study the actual chemistry, we use the methods of QMMM that Alessandra very nicely introduced to you the other day. And here we run very often with hybrid functionals, and it can be basically plain MD simulations on a QMMM level, or then free energy calculations where you can use something like umbrella sampling to study this. And for this, I don't have the time to go into the details, but we have to introduce some kind of collective variables, for example, on how the proton would move along this type of water wire in the system. And what you can usually do at density functional level is to sample, let's say in an umbrella sampling, some picoseconds per window, which is not very much, but of course, you can then supplement the thing with a classical sampling of the system to get representative ideas of the dynamics. What we also favor very much in my group is to use density functional theory-based cluster models, where you try to represent the key chemistry for the system. And why I like this type of approaches is that you can also study the system very well with ab initio methods and try to do, so to say, good electronic structure calculations. And we have started using, for example, the random phase approximations in recent years quite a lot to look at these processes. And very often, this is, for example, a free energy barrier calculated with QMMM or with DFT, you get not identical, but at least qualitatively similar pictures, which is, after all, what you're after to get the mechanistic understanding. So basically what we then try to do is to model the, so to say, catalytic cycle of the system by running dynamic simulations, injecting electrons into the system, and see how of this. Very good. I seem to have some problems with my internet connection, but interrupt me if you can't hear me well. So let's have a focus on what happens in the machinery. And the first realization that we have had over the years, we and other people working in the field, is that the proton pumping process is activated by the quinone reduction process when electrons start coming into the quinone molecule. And of course, to be able to treat this, you need to have the QMMM approach. And this I introduced to you a little bit before. But the other thing that you need to think about is how to model electrons, which is not fully trivial when you have systems like iron sulfur centers, because each iron can carry either 4 or 5 spins, which you can then couple antiferromagnetically to 6 unique combinations. So this is basic combinatorics. But why this matters is that you can have big energy differences between the unique spin states, so up to 5-6 kcals per mole. So it is rather important to prepare the system in the chemically correct state. And this cannot be taken into account purely by electrostatic effects. And then we need to have ways to drag the electron, put the electron where we want, for example, on the metal center or on the quinone center, and transfer it between the different sites and look at the process and what it leads to. So this is the result of such a simulation. So basically you can see here the spin density disappearing from the quinone site. So basically there is an electron motion, and this couples to movement of some nuclei, some protons together with this. So if I zoom into the quinone center, you can possibly see on your screen that when the electrons come in, you basically see that protons are being pulled from nearby residues to this quinone molecule, forming effectively quinone. So we have a tyrosine and a histidine residue, and upon reduction, we basically form this type of species. And at the same time, because of the charge relocation, you basically break or weaken this ion pair between the histidine and an aspartate. So with QMMM calculation, you can look at the system only on a very limited time scale, but in order to see what happens on longer time scale, you can run classical atomistic simulation, and we discovered something quite interesting. So prior to quinone reduction, here is zooming into the membrane domain. You see basically that there are, here is this aspartate at the active site and some other carboxylate at the site at the membrane domain. And basically upon quinol formation, you break this ion pair, which leads to some conformational changes of charge groups here in the membrane. So it's kind of interesting that you have this consecutive plus minus charges organized kind of in a wire. So if you tickle the first, you get a response kind of at the end. And this basically leads to big pKa shifts that can be indicative of a proton uptake reaction. So how could we test that is this relevant for the real system? And one idea is, of course, that you can kill some of these charge residues and do a mutagenesis experiment experimentally. And this is what we did. So we reconstituted the complex 1 in proteoliposomes, gave electrons with NADH. And basically, then we could measure how protons are being pumped inside these proteoliposome vesicles by having a pH or in this case, a charge-sensitive dye. And you can see that there is a big difference in these traces between the wild-type system and when we have removed this charge in the system. And what's quite interesting is that if you look at the other residues involved in this charge communication, is that many have a very big effect on the activity. And this is also seen to be a site-related, a hotspot for human disease-related mutations. Good. So we have formed now quinol. And then what we think is happening is that the quinol motion out from the cavity is giving the energetic kick for the proton pump. And the reason for this is that when we look at the quinone potential, redox potentials in the active site, it has a very low potential around -300 millivolts or so. And which means that when it gets electrons from an electron donor, for example, NADH, it is almost equipotential and there is no energy being released. However, quinones, when they're in membranes, are much higher potentials, which means that when you go from this -300 millivolts to +90 millivolts, you have a release of the energy. So already thermodynamically, this must be the kick. So what we did is that we wanted to study this process further, and you can see quite nicely. So this is we studied the process with free energy calculation. Here is now an umbrella sampling simulation where we basically started identifying local minimas in this site. And the first one is just the site I showed you before, but then there's some very interesting structure in this. So this is basically how the quinone moves out of its cavity towards the outside. You see a local minima that matches quite nicely with an inhibitor-bound form of this enzyme. And then you have here quite much further out in the membrane part of the system, two very, very interesting local minima. And we think that these are important stalling sites for the quinone. And what is quite nice is that we were recently able to resolve this second binding site by cryo-EM, where we can see an inhibitor binding to this predicted site. So I think that it provides a very nice validation to our free energy profiles. Okay, so we have produced quinol. We have moved it to a second binding site, which is located here in the membrane. And now we would like to understand how does that trigger the proton pumping process across the membrane. And when we run MD simulations, there is something quite interesting happening. And now I show you just the kind of side view of the system, and you can see that during the dynamic simulation, you have a formation of these water wires at various places. And just if I would show you here a time average picture of them, you can see that they start forming at various sites, basically 4 specific regions in the protein. And this seems to be similar for bacterial systems, for when we do simulation on the much larger mammalian complex 1, but also a photosynthetic variant of the complex 1. And these water wires form basically at symmetry-related locations. These form at antiporter-like subunits, and basically you have an internal symmetry such that they form around a broken helix, where the input and output channel are related by symmetry to each other. So if you would flip this 180 degrees and by a rotation, the input and output channel come to the similar location. And what controls this hydration process is a charge residue at the end of this broken helix. And you can see that upon hydration, there is a small subtle motion of this broken helix element, which I guess is why it's a broken helix element. And you can see that if you then during the simulation deprotonate this residue, then basically you get de-wetting of the channel as well. So we think that the charge at the bottom of this creates an electric field, and that pulls in water dipoles, so to minimize the energy. And basically when you do not have this closes the gate. So you could say that it's a little bit like a field effect transistor where the gating charge regulates conduction properties of the device. So this is, of course, we've seen similar behavior in many other systems, not only in complex 1, but in cytochrome C oxidase, where a reduction event causes a similar wetting behavior, but also in light-driven systems like in this sodium pumping rhodopsin, where the charge changes causes differences in hydration across the membrane. So now the interesting question, how are these proton transfer reactions coupled to each other? And there is a very interesting internal symmetry in the system. And this is basically that you have this charge residue, this central lysine residue, which I talked about, that regulates the hydration state of the channel. And then basically before that, you have close proximity an ion pair. And when this ion pair is in a closed state, this lysine prefers to be in a protonated state. But we can also see that there is another possible state where this ion pair can open up, can dissociate and shift and form contact with the previous subunit. And this state becomes, of course, favored when this lysine proton residue gets protonated, and that is to minimize the charge repulsion between the groups. And this you can see quite nicely in free energy profiles, in classical free energy profiles. But this also affects very strongly the proton transfer energetics itself. So you can see that when the ion pair is open, you have quite enough high barriers and endergonic proton transfer processes. And when you open up this ion pair, the proton would like to go. And this is basically how the proton motion would happen. It goes via these water molecules to an intermediate histidine residue, and then from the histidine residue, it continues onwards to the terminal residue over here. And we have some snapshots where we can see this even spontaneously happening in QMM and MD simulations when the state is open. So you saw maybe this histidine residue here that is on the pathway. So it's kind of interesting that it's basically an important stalling site. So we thought that what if we mutate that? What if we take it away and introduce there an alanine residue? And we did the same pumping experiments from here. And what you can see is that you see a very drastic decrease of the pumping effect by introducing this mutation, but you can also see a quite significant change in the quinone oxidative reductase activity. And this is quite interesting because the quinone site for this subunit that we studied here is over 100 angstroms far away. So it's a true coupling effect in that sense. What is also, it fascinates me really in this system, and I think it's general for many other systems, is that, of course, the water molecules are key for transferring the proton. Without them, you would have too long gaps to have an effective proton transfer barrier. But water molecules are also key into causing the proper dissociation dynamics of this ion pair. So only when the channel is in a wet state, you basically can open up this ion pair with some reasonable energetics while you have a very high barrier in the dry state of the channel, which I think makes sense also from an electrostatic perspective. So we could put all of this together basically to say that the quinone reduction chemistry by these local residues, it activates the proton pumping process. The quinone has a second binding site in the membrane domain, which brings it closer to the proton pumping subunits. And then we have this long-range signal propagation processes, which take place by a channel hydration process, by ion pair dynamics, and then by this coupled proton transfer process. And of course, the proton transfer wouldn't be possible without controlling the hydration state of the system. So we can draw up a mechanistic picture based on this. So electron transfer to quinone produces a quinone molecule. The quinone moves in to the second binding site. This initiates the proton transport cascade, moving the proton along this membrane domain close to the first ion pair. The first ion pair dissociates because that is, you get accumulation of charge. The dissociated ion pair pushes the proton laterally to this end of this yellow subunit. It opens up the next ion pair. You push the proton, you open up the ion pair, and you have pushed the proton to the lateral end. Now there is no more neighbors to transfer the proton. So what happens is that the proton is most likely going to leave the system. This site is also very close to the exit site, at least in our MD simulation. And when the site leaves, there is a pKa increase of the central lysine residue. Uptake of this proton would close again the ion pair, but this in turn destabilizes the proton loaded here at the interface of these two subunits. So the proton is being pushed outwards in here. You uptake a new proton, you close the ion pair, you release the proton, you take up a proton from this yellow subunit, close the ion pair, and release the last proton across the membrane. So it kind of transfers, so to say, back the signal, which is then coupled to the release, or that is what we have proposed. And then basically the quinone needs to be replaced by a fresh quinone to restart the engine. So with these things that I've showed to you includes hydration changes, some subtle motions in helices and other things in other structural elements, and changes in hydration state. But there is also a very interesting larger scale conformational changes involved in this machinery that I thought could be interesting also for this community. So when we run MD simulations on this system, based on the cryo-EM resolved structures, what you can do is that you can look at the global motion of this system, and you can group them together by PCA or other type of analysis, and you can see that there are two types of motion. There is basically a bending motion of this machinery, and then there is a twisting motion as well. And these are quite large-scale motions happening during the dynamics. And what is quite fascinating is that if you compare these motions taken from simulations to what is being resolved in cryo-EM particle classification, is that you have exactly similar type of motions in those two states. And in the cryo-EM, these two states, these two kind of motions have been linked to the active and deactive form of this enzyme. And what happens then if you drag the system along this twisting and bending motion, these are large scale motions, so very rare events, and you need to apply special techniques for this, of course. You see that you have a strain effect accumulating at very specific locations of the subunits. And at some of these locations, there's been quite recently observed conformational changes in certain side chain positions. And we think that these are also very important. And one process where we can see this coming into action is basically when the quinone moves upwards and downwards its channel. So in this process, complex 1 seems to undergo first a twisting mode and then a bending change. So there is also larger scale things happening, whether it's the cause of the process enabling the electrostatics or vice versa. We cannot, of course, comment at this point, but I think that it's important not to forget about those events as well. Alessandra, do I have a time to show one last quirk, or is my time up?
Yeah, sorry.
My time is up for the system. That's totally fine. So I think that I had time to introduce to you the important thing, maybe just out of interest, and you can look at the paper. We resolved a fascinating variant of the system where new modules have been placed in, and this is used by cyanobacteria to actually concentrate CO2. And this is a very, very important process in photosynthesis. And I think it's fascinating that you use basically the mitochondrial enzyme to do this. And this is I think a quite nice interplay between cryo-EM structure resolving and simulations, and then some biochemical work. So I won't show you this. You can look at the paper, but I would like to maybe put a few summary points of the thing. So I hope that I convince you that by integrating computational approaches with maybe biophysical techniques, you can get a very powerful way to ask mechanistic questions of the system on a very broad range of timescales and spatial resolutions. I described to you how we think this long-range redox signal propagates around this system. I showed you some evidence for the substrate chemistry, how we think it happens, takes place in the system. And then I presented to you a mechanism that involved both forward and backward propagation of a pulse in this system. And with this, I put the group picture. This is where you can see that we are organized in the shape of complex 1 over here. These are the people from my group, and also some previous members, and some funding agencies. So I'm happy to answer any questions you might have of this presentation. Thank you so much.
Thank you very much, Ville, for this very nice and interesting presentation. Questions? So I start maybe with a question myself. Can you a bit comment on how you model the electron transfer process that you showed at the beginning?
Yeah, right. So what I showed here is involves the very last step of the electron transfer process. And what we've done there is basically various type of models. So one is that you shoot basically the system, start looking at the dynamics by QMMM simulations. And then there is in these states that we have studied, there's such a big driving force that the electron want to combine, and we can actually see spontaneous combination of the protons and electrons in the system. Okay. And what's kind of cool is that it's reversible. We can also see in trajectories the reverse mode of the protons going the vice versa, and the electron jumping back to the electron acceptor. Okay. This is one way of studying the system. But then what we have also done is to go in a more, so to say, classical way of looking at the electron transfer, trying to determine redox potentials, reorganization energies, coupling elements, more in a traditional, maybe quantum chemical approach, which would then allow you, using some type of, let's say, Marcus theory, to get an insight in what would be the time scale for the overall electron transfer event. And I think that is a very interesting question by itself. But from the mechanistic point of view, what triggers the thing is when the electron reaches the quinone, right? The quinone. So I think that that's of special interest. But then looking at the long-range electron transfer process is, of course, pretty cool as well, I think.
Okay. So don't be shy. There are lots of compliments, including one. Thanks a lot, sir, for the very epic presentation.
Excellent, thank you.
Hi, could I ask a question?
Sure.
Yeah, thanks for a very nice talk. I'm just wondering how the sort of the degrees of freedom of the membrane couple into this, because it's a membrane embedded protein. So do you get a lot of like perturbations from the lipid molecules around the protein?
Thank you for asking that question. So it is, in fact, I wonder, can I do I have a spare slide on this? So the membrane is extremely important for this process. And it is, in fact, so, let me see, do I have it here? That one particular lipid is of key here. Can you see my presentation mode? Yeah. And that is cardiolipin. And it seems that cardiolipin, what it does is, this is coarse-grained because we can't address these on a full atomistic timescale in how they associate. We have done atomistic simulations also in here. You can see that cardiolipin associates with very particular places in the system, and they seem to match cryo-EM structures quite well. But what this cardiolipin does is that it actually affects the motion, these global motions, in exactly these twisting, bending motions. And it is such that this is the slide that happens the twisting and bending, coordinated twisting bending happens when we have cardiolipin in the system. They're very coordinated. We don't understand exactly how, but somehow the cardiolipin affects this. And when we remove cardiolipin from the system, let me see if I'm here, you can see they're all over the place. Yeah. And this is data from one millisecond of yes, coarse grain simulations, but anyway, so we have quite a lot sampling in here. So you're absolutely right that the lipid molecules are somehow very, very important, at least for the global motion of this system. Did this elaborate? Does this answer your question?
Now there is a question from the audience, from the chat. Can you please explain more about the interaction between electron and proton and phonon?
Yes, or right, protons in these cases, right? So basically the electron-proton coupling, I mean, it happens, so to say, at two specific locations where they combine to the same chemical group. And that is first at the top site, the NADH site, NADH-FMN, and there you basically have reduction couples to protonation event. And the second site is at the quinone to quinol, right? So two electrons react with the quinone and two protons are needed. And that basically forms them together. Then all after that is basically proton transfer processes. So these local charge shifts induces conformational changes that increases the pKa values, changes the hydration state of the process, and then that triggers in turn this protonation cascade. So then you have basically decoupled the protons and electrons. They don't move to the same sites anymore, but they're tightly coupled, right? And that you could see, for example, by this mutation experiment. You do a mutation 100 angstroms away from the groups involved in proton transfer, you can see a change in the activity, meaning how electrons come into the system. So there's the thermodynamic coupling there, which is extremely tight.
Oh, another question. I have a little question about the ion pair. Why is the energy profile with the dry and wet cases not symmetric around the D0 point?
No. This is a question, energy. Yes, dry and wet cases. There is a shift. So I think that this is related to what is our reaction coordinate, right? It's composed of a distance difference between different sites. So I think that this shift basically in distances is related to how we have defined the ion pair. But the asymmetry, of course, is partially also due to the fact that when we have a hydration, it's not exactly anymore the same structure. You could see, for example, that some helices move. There are some conformational changes. And that is also why some distances between the groups, in fact, are not anymore the same in the wet and the dry state. And I think that this probably relates to also some local asymmetry in the free energy profiles.
Okay. Thank you very much. I think there are no more questions. Thanks for joining this event and for the nice presentation again.
Thank you so much for inviting me. Thank you.
Welcome. So we have the afternoon session at 2 p.m., sorry, and we reconvene at that time. Thank you again, Ville. Bye.